All right, I have a little bit afternoon Pacific time, so I think we'll go ahead and get going. Welcome everyone to the PacVec Spring Seminar Series. Thank you for taking time out of what I'm sure is a busy schedule. Today we're going to have two speakers, so I'm just going to give quick introductions, though I'll leave most of the introducing to the speakers. We'll have time for Q&A between the talks, so the first speaker will talk for about 20 to 25 minutes, a little time for Q&A. Second speaker will talk then with a time for Q&A at the end. So our two speakers both are from UC Davis today. First will be CC Edwards. CC got her bachelor's degree at Baylor University. She got her master's at Texas Tech, and now, as I mentioned, she's at UC Davis as a PhD candidate. And today she's going to be talking about her research looking at pyrethroid resistance. And our second speaker, I'm just going to have you say your name out loud for me so that I don't mess it up. Oh, I think you're still muted.
I'm still muted. Ania Kawiecki.
Ania Kawiecki, thank you.
Yeah.
Um, Anya is a veterinarian. She has her DMV and her DVM, excuse me, and she's currently training to be a research entomologist or a research epidemiologist also at UC Davis. Okay, I will hand it over to Cece. Like I said, she'll have 20 to 25 minutes and then we'll pause for questions.
All right, thank you so much for that introduction. Can you guys see my screen? That's always the, all right, you guys can see my PowerPoint, thumbs up. Okay, awesome. So yeah, as I was introduced, I'm a first year PhD student here at UC Davis, and I study resisting resistance. So identifying biochemical biomarkers for pyrethroid resistance. I'm a first year PhD student in the lab of Dr. Jeff Attardo here in the Department of Entomology, and I am a PacVec trainee. So to introduce the Aedes aegypti. So for those who don't know, my advisor is Jeff Attardo, and he's well known for his photography. And I just love these images that Jeff has because they just show that we think of mosquitoes, we think of they're ugly, but I think they're pretty up close if you look at it. But this is the Aedes aegypti, also known as the yellow fever mosquito. And in the United States, it was introduced around the 1700s, at least in the southern United States. And they are predominantly the largest contributor to arboviruses worldwide. So dengue, chikungunya, and the Zika. But in California, it was pretty recent when Aedes aegypti became introduced. So originally, I'm from Texas, and so we always had Aedes aegypti there. And so coming to California, not realizing what a big issue these mosquitoes are. So for reference, in 2013, they had an occurrence of Aedes aegypti in the Central Valley. And as the last decade has expanded on and on, we see this mosquito makes its way from Southern California into Central Valley. And here where I am at UC Davis, for those who don't know, I'm near Sacramento, up north. We're now seeing occurrences of Aedes aegypti here. And it is an issue that we're seeing these mosquitoes. And it's also not just an issue in California, but neighboring states. So there was an outbreak of Aedes aegypti in Las Vegas last year. And when they did genetic analysis, it came from California. We do genetic analysis of the mosquitoes here in Northern California, and they can trace back to Southern California. And a lot of neighboring states are now showing Aedes aegypti. They reproduce heavily, and it's a huge issue. And with that, the problem is pyrethroids is a common insecticide worldwide. They use it in California, they use it worldwide, and insecticide resistance is an ongoing concern. However, little is known about the biochemical metabolism mechanisms associated with insecticide resistance, which is what my lab and my research focuses on. So a little bit about pyrethroid resistance. How does a mosquito become resistant to begin with? Well, there's a target site. So here is a sodium, the voltage-gauged sodium channel, and that can be modified. So it could reduce and eliminate the pesticide effects through like point mutations. Also, we have here the cuticle thickening. So the cuticle, kind of think of it as the skin of the mosquito. So over time, we can have overexpression or another point mutation that causes the penetration. So it's slower penetration to the mosquito overall. So the mosquito is resistance. And then what I study and focus on is metabolites and metabolic resistance from detoxification and sequestration of the mosquito. Resistant insects have higher levels of enzymes that can break down insecticides and can get rid faster of these toxic molecules. And these enzymes are efficient and just over time can degrade. And with that, metabolic resistance is one of the most common mechanisms of resistance. And with that is the greatest challenge we face, and challenges these insects as they can break down these insecticide faster. So what do we do at our end with it? And that's what my research focuses on. And so why is it important to study insecticide resistance overall? Like, I'm not saying that we need to figure out and solve insecticide resistance to spray more insecticides, but more just that understanding insecticide use helps understand insect biology better and mosquito biology. So external factors can affect internal factors of mosquitoes, and which shows overall vector competence, which is these mosquitoes also spread viruses, which is detrimental to human health. So understanding insecticide use also helps us understand mosquito biology. And just to think about, like, if we think about humans, if we were exposed to toxins and poisons all the time, we would potentially not survive as long as mosquitoes do. And so I think it's really cool just understanding the physiological components of how do these mosquitoes become so resilient and under all of these pressures. So with that, why is it important to continue is, well, we're having some outbreaks of dengue in the Western Hemisphere. So in Puerto Rico, they declared an emergency from the CDC, and there's been multiple over millions of cases in Latin America, and you can see a person is fumigating here. In order to help the control mosquitoes. But then you probably think, oh, well, the US, we're in mainland United States, we're immune to it. Well, no, we're not. We have the mosquitoes here. So there's been locally acquired cases of dengue in Texas and Florida. And last year we saw locally acquired cases of dengue. And so with that, insecticide resistance is an issue. And with these outbreaks, they're showing that these mosquitoes are resistant. So this was response after the 2016 outbreak, but they were having an issue, but they later realized that these mosquitoes were resistant to pyrethroids when the outbreak happened in 2016. And oftentimes we kind of find out later than sooner when mosquitoes are resistant to pyrethroids. So it's better to understand insecticide mechanisms, understand early detection of resistance, which my research will talk about, and just understanding overall why is it, to help understand better ways to detect resistance and also how to respond to that. So that's what my overall research is what are new ways we can resist resistance? What can metabolomics tell us about insecticide resistance? And can biochemical biomarkers be used to monitor pyrethroid resistance? So flashback to high school or freshman biology, we have the central dogma of DNA so we have DNA becomes RNA, which becomes protein in the biochemical and the phenotype of the mosquito. So with that, a lot of insecticide research focuses usually either on the phenotype, which you probably heard CDC bottle assays, which shows if an mosquito is resistant to a certain insecticide, or we have DNA. So we have this this gene called KDR, which we can use to detect a genetic resistance. But if you look, we have RNA, protein, and the biochemical response. And so there's a lot of different things happening between these two responses. So with that, my lab focuses on the metabolome, which is the biochemical response. And with that, the biochemical response is the closest to the phenotype. So through metabolomics, we can look at chemical compounds that is the closest to the phenotype before we actually see the phenotypic response of resistance within a mosquito. So what did we do to help determine metabolomics? So we got some mosquitoes from the Central Valley, which is Clovis, California. Those, just think about California, you throw a dart in the middle of California, that's where Clovis is. And we did the Rockefeller strain, which is a common mosquito lab strain. So what we did is we exposed this Central Valley strain of mosquitoes with insecticides. So we use pyrethroids, and we exposed them for 3 generations. After that, we sent them for metabolomics. So here at UC Davis, we have the NIH West Coast Metabolomics Center. The control, the Rockefeller strain, was just treated as normal, not exposed to any insecticides at all. And they were matched with 5 days post-eclosion. They were sugar-starved, and they had the same larval density and diet throughout. So with that, when we got our results back, looking at the PCA, principal component analyses, we here have our primary metabolites, which is going to be the Clovis strain, which is our resistant strains, is going to be represented by the purple, and the Rockefeller, which could be our control, is going to be represented by the green. So when we look at primary metabolites, we can see that the Rockefeller strain has metabolites that are closely related to each other, that are separated from the Clovis strain, and same thing with lipids. So when we do a lipid and metabolite analyses, you can see that the Rockefeller strain is close together and clustered, but when we do a second least squares analysis, we see more separation between the primary metabolites between the Rockefeller and the Clovis strain. So you can see they are specific metabolites associated from each mosquito that we weren't seeing in the other treatments and control. And we can see that as well with the lipids, that there were certain grouping of lipids that we did not see, we saw in the Clovis that we did not see in the Rockefeller. And from there, when we look at a metabolite analysis within, when we do a heat map, we see between the Rockefeller and the Clovis, you can see the red represents that there is more of this metabolite, and the blue represents that there's less. So we can see that we have higher levels of sugar, glucose, fructose, or hallulose. And these are components, again, flash back to your intro bio courses of components of the PPP, which is the penthose phosphate pathway. An overview of that pathway is that there are functions of NADPH, and overall we have a response. And currently we're working on a synergist to help with this, which I'll talk more about, using this glucose 6-phosphate dehydrogenase. And so looking forward, when we did a sample clustering of PCA. So this is a slightly different experiment from the previous one, where I showed you earlier that we sent them 3 generations of insecticides exposure, and we didn't do any more exposure. This was only 1 generation of insecticide exposure. So we exposed them with insecticides from LA and Central Valley, then sent them for metabolites. Metabolomics. So just to clarify, with that, we can see here we have Los Angeles, so LA, which is represented by the dark blue is resistant and the light blue is susceptible, so non-resistant. And then we have the Central Valley, which is this light orange yellow color, and then this dark reddish color is going to be the susceptible, and the light is going to be the resistance. So when you look at individually from LA and Central Valley, you can see the clustering of metabolites specific to each one. And then when we look at the Central Valley, you can also see clustering. There is a little bit of overlap, but there is individual. And when we look at all 2 of them together, you can see that there are the resistant strains do have clustering together with similar metabolites, and then the susceptible are separate. But when we look into further analyses, 48% of these metabolites differ between the resistant and susceptible exposed samples. And with that, biomarker analyses shows that we have about 50 metabolites that can be a reliable classifiable status to help detect insecticide resistance with pyrethroids. So what's next with that? So this is going on to what my research focuses on. Can biochemical biomarkers be used to monitor pyrethroid resistance? And can lower costs biochemical biomarkers for humans be used on mosquitoes to monitor pyrethroid resistance? So kind of like something to think about is the PPP pathway, the pentose phosphate pathway. It's the same in humans as it is in mosquitoes. Biologically, we kind of think insects are animals, so there's already biochemical tests established for humans. So my research is focusing on, can these biochemical tests meant for humans be used on mosquitoes? And that's what I'm currently doing. So these are some of the punitive biochemical biomarkers I've been working on. So if you look at a few of these, so LPE, I'll go down a list of a few of those. So these two, LPE is the lipo, so LPE, so let's start with pyroxalic acid, helps break down B6, B12, sorry, B12, and then LPE, which is a lipo protein lipid enzyme, helps remove fatty acids. This one, LPC, it's a mouthful, lipofosphatidyl chloride. It helps break down fatty acids. And then you see some of these tyronine, nicotinic acid also break down B vitamins. And then you probably see like supplements for humans, right? Like histamine, you think of allergies, but it's also a regulatory component for mosquitoes. Biotin as well helps regulate and signal cells activity in the mosquitoes. And then adenosine is also a regulatory component in mosquitoes. And so when we looked at these specific tests, some of these, depending on the Los Angeles, we had the Los Angeles, the Central Valley, and the Rockefeller strain versus Clovis strain. But if you looked at the susceptible and resistant populations, some of these had a fold change. So the red showing that there was more of this component. So like we would see a higher component of changes in the resistance versus the susceptible, and then the blue ones showing that we would show more on the susceptible rather than the resistance. So these biochemical tests will go either way. And so this is matched on the right here of what biochemical assay I'll be using to help determine which one of these work. And as of now, I've only been here for what, 7 months, 8 months. So most of my time here has been modifying using these biochemical assays as it tells you to use human tissue, like swab someone's cheek, but you're like, with a mosquito, you can't really do that. So modifying these tests these last few months to help change it, have to do some dilutions and modify these tests to show it working for mosquitoes. And I've done that. And now I currently I'm working on 2 tests at the moment. And I am showing differences. However, still working on that and didn't want to present data today that wasn't 100% ready to show. But with that, going on of what I'm actually doing. So previously I showed you that recently we used the Clovis versus the Rockefeller strain, but we got feedback from previous publications that the Rockefeller, it is a very inbred strain and it's very commonly used. So we wanted something that was more found in nature. So we used the Clovis strain, Central Valley strain. So what we did is we grabbed some mosquitoes from the Central Valley, and currently also remaining colonies. So I currently have resistant strains, so strains that I am pressuring resistance by spraying insecticides every generation, and some of them die. The ones that survive, they reproduce and lay eggs. And then the susceptible, again, they have no resistance whatsoever. And I'm currently on generation 7 for these biochemical tests. So they are used in evaluation to compare the 2 groups together. So they are from the same genetic background. And they're also each of these there are 3 groups within each treatment. So I have 3 groups of insects that were collected that are resistant and 3 groups that are susceptible to show genetic differences between different group because they're they're from different pools, but they're genetically from the same location. And with that, some money we received from PacVec helped. So the cool thing about these tests is you only need an easy spectrometer. You don't need something really large, but we got this kit, so I've been able to use because all you need is to measure absorbance or fluorescence, which you can easily do with some spectrometers. But we got this huge Flu-Star Omega machine, which can help. I think we can do about 300 wells at once. So to help establish this data set, it's been really easy and really convenient to have a machine to help establish a lot of tests all at once. And with future directions, so I am working on that, but what is next? So future directions is these biochemical tests in semi-field conditions. So as you know, something can work in the lab, but may not work in the natural environment of mosquitoes. So I'll be driving down to Clovis and this summer, or not Clovis, but around the Central Valley, and running semi-field conditions where I get wild mosquitoes of Aedes aegypti, put them in cages, and test them before and after pyrethroid spraying. So they're going to be doing the insecticide spraying. They only do it twice a year, so it's very time sensitive to get in there and collect the mosquitoes before and after to show that these biochemical tests do show resistance after exposure. And then also looking at potential synergists based on the metabolomic data. So as I talked about, some of these inhibitors of these pathways, if we inhibit some of these enzymes, would this potentially make resistant mosquitoes susceptible again? And so we have a few, we did some preliminary data, and there are a few synergists that a previous student did before me, and it seemed to work. And so we have a list of 3 potential synergists that I'll be establishing for my next aims of research. And then also we got a Raman spectrometer, which basically does, we're getting it approved and bringing it to the lab. So what it does, it kind of tells you the metabolite data all at once. It's this really cool machine, and it'd be cool to get some resistant mosquitoes and put them through the machine and see if the metabolites associated are the same as the biochemical test, but also messing around, like, do we need a whole mosquito, maybe mosquito frass, or can a mosquito leg or like something small tell us more about the metabolomic profile of the mosquito, which I think is a really cool. I'm excited when we get this machine this summer, and I'm probably going to be, I don't want to say playing with it, but kind of messing around. So in conclusion, the Aedes aegypti and insecticide resistance is a big concern in California, where we're focused, but worldwide, as we can see with this prevalence of dengue outbreaks. The metabolomics does show significant differences in biochemical profiles of susceptible versus resistant mosquitoes. And ongoing research is currently investigating if manufacturer biomarker bioassay tests can help detect these differences. So with that, thank you so much for presenting the opportunity to talk. I would like to thank my lab members. So Dr. Taylor Kelly and Dr. Lindsay recently graduated, but their research laid the foundation for me to carry on. And Nicole is my lab mate. And this is done with a bunch of other abatement districts and people that are, some of them are funded by PacVec, but they also are all curious and wondering what we do about Aedes aegypti here in California. And of course, PacVec for providing me the training grant and the funds necessary in order to present and complete this research. So with further ado, I'll take any questions.
Great. Thank you, Cece. Nice job. Any questions? I can get us warmed up with one. So I understand the concerns about Rockefeller, right? I work with Tarsalis mostly, not with Aedes aegypti, but I know that there are some issues with detoxifying enzymes and other things in the colonies. I'm wondering, so you're collecting from the field and then you're selecting for resistance. How susceptible is the group that you're calling susceptible?
They're not really susceptible, right? We can argue that no Aedes out there is really susceptible if you think about it, right? So that is something that I have talked about, but I am doing LC50s with both groups. So within the 2 groups, like the quote unquote susceptible, I did an LC50 this weekend actually. So there is a few changes, like I think within like 0.1 milligrams per milliliter of the LC50 changes. So they are less resistance, I would say, than the resistant mosquitoes. But if I was going to compare it to like an LC50 of Rockefeller, like it probably takes like very, very small amount to kill a Rockefeller with a permethrin. So Yeah, so we can argue, in this case, it's more of just that they're less resistant to show that differences. However, it is more of, I say, accurate of what we would be seeing in nature than what Rockefeller would be, because Rockefeller is just, I don't want to say a lab mouse, but it is very a lab grade mosquito that does not accurately depict what we see in the real world.
Other questions for Cece? Feel free to just unmute and ask. It's not a huge group. I can see everybody on my screen. All right, I think I've taken the sufficient pause. Thank you again, Cece. Nice job.
Thank you.
All right, moving on to our second speaker. Take it away, Ania.
I'm going to share my screen. Let's see. Oop. Where is this PowerPoint? Oops. This is the end. Sorry. I should have been, let me stop sharing for a second. Sorry for that.
No problem.
I was just working on it right before this started. Okay.
All right.
Can everyone see that? Okay, perfect. Sorry. Okay, so I'm Ania Kawiecki. I'm a student in Chris Barker's lab. I'm actually, I think, on the other stage as CC, so I'm like finishing my dissertation. And this project that I'm going to talk about is actually accepted for publication, which is great. So I'm going to talk about the spatial and temporal analysis of the impact of ultra low volume, which I'm just going to call ULV from now on, indoor insecticide spraying on Aedes aegypti household density. So actually, Cece gave a really great introduction, I think, to Aedes aegypti, also mentioned the huge problems we have with dengue, especially this year, I think is really, really bad. But so Aedes aegypti, as we know, transmits many arboviruses, including dengue, but also Zika and chikungunya. And ULV is a pretty commonly used method, especially in these endemic countries where dengue is so prevalent, to try and cut the transmission cycle by reducing the number of mosquitoes. And it is actually often very used indoors as opposed to outdoors, which I think is more common in contexts like the US. So what my well, and this is just like a reminder that this is a life cycle of Aedes aegypti, so it has a water stage with some eggs, larvae, and pupae, which is not affected by these insecticide sprays. They only affect the adults. So even after there's a spray, there can still be reemergence from untouched or unaffected water sites. So the questions that I am asking in this project are basically, how long does the effect of this spray inside a household last? So for how long can we see a reduction in the numbers of Aedes aegypti inside a household after one of these sprays occurs? And also, like, is there an effect in a given household of sprays occurring in households that are surrounding this household? So imagine you're this household in the middle, the one in the black outline, and at a given day, maybe this household was sprayed the day before, but on the next day, its neighboring households are being sprayed on that same day. So which one matters more, the spray occurring in the household itself or the sprays occurring around the household, and what is the temporal importance also, like how long does a spray actually have an effect for? So these are the questions. And so the way I tried to answer them was using data from two studies that occurred in Iquitos, Peru, which is in the Peruvian Amazon, where dengue cycles continuously. It's endemic there. And this is kind of what a neighborhood in Iquitos might look like. So you have more like the urban areas where houses are row houses that have like street facing doors and windows, and also in the other end patio. They're kind of like what we call here shotgun houses. So they're like narrow and also just like right next to each other. And then the other extreme is like these houses on the river. But in Iquitos, it's very common. Most of the mosquitoes are just indoors. There isn't like a ton of outdoor space per household for the mosquitoes to be. So indoors is really where the focusing of the control happens. And so how these studies occurred, there was adult aspirations before, during, and after experimental cycles of indoor insecticide spraying with permethrin. And there were 6 cycles that occurred like once a week. And so we have data on the number of mosquitoes before, during, and after, and collections occurred like every week or something. And this data was already analyzed in a paper that was by Gunnings that was published in 2018. But in that paper, they were looking more at, so the way the studies were implemented was there was a central spray area, and then there was a buffer area around, and mosquitoes were being collected in both areas at all time points. And so they were comparing like the effect of sprays in the spray area versus a buffer area. Whereas what I'm trying to elucidate is more fine-tuning the effect on individual households of a spray and of the sprays in the surrounding household. So a little more spatially explicit. And so the way the study kind of worked was I had this data from these 2 studies, and then I developed an array of different models that measured in different ways the effect of sprays within an individual household based on how long ago a spray had occurred in that household. And in addition, I also looked at models that include an additional term for sprays occurring in the surrounding households, and I'll explain later how this worked. And then I compared these models and selected the best one. And that allowed me to also estimate kind of the recovery curves of mosquitoes after a spray. So, excuse me. So my first question, is there a cumulative effect of multiple sprays on the same household over time? So basically this is trying to answer, if your house was sprayed yesterday, does it matter if it was also sprayed last week, or does it just matter if it was sprayed yesterday? And so the way, I mean, I don't know if this is useful, but the way I answered this was I calculated using all these different decay curves. So according to all these different decay curves, I assigned a weight to when the spray had occurred in that household. And I tested models that assigned the weight according to only the most recent spray, or like a cumulative weight of all the sprays that had occurred. So that would be like a higher number than if you're only looking at the most recent spray. And what I saw from comparing these models and their fit is that really the models that included a weight of only the most recent spray fit better than the ones that included this cumulative weight. So that indicates to me that there is really no improvement in the model by including this cumulative weight. So really what matters is the most recent spray. And so that leads us to the next question: how long does the spray effect actually last over time? And so when we look at the best fitting models that are best fitting among the 2 studies, because the 2 studies occurred in 2 different years, they had a little different study designs and particularities. So overall, the best fit was kind of in this area. These are the best fitting models. And of those, we chose this consensus model, which is this bright red one, which was assigning weights to days since most recent spray using a Gaussian decay curve, which is this, with a specific sigma. And so this is the consensus model that I called M-best. And using this model, I was able to develop also this, it's kind of the same, just like it's like the interpretation is a little different, looking at how much reduction of Aedes aegypti can we expect with the days having passed from the most recent spray. So really the day of the spray, we see around a 50% reduction in Aedes aegypti in a household. And then that effect wanes over time, where there's half that effect after 25 days, and after 50 days, there's no effect at all. And the last question that I was asking in this project was this more spatial question. So if you're this house, and maybe on that day you're not being sprayed, but two of your neighbors are being sprayed on that day, but also maybe yesterday other neighbors were sprayed, and that was like pretty close in time as well. So are these sprays occurring around you actually affecting the number of mosquitoes in your house? And I similarly compared an array of models that included an effect that was measuring this in different ways. So I used like a more simple way of calculating this, which was just percent of houses sprayed in the past week within a given ring from each house. So in the 100 to 200 meter ring around a house, what percentage of houses were sprayed in the past week? And that's like a crude way of measuring this. And then I also did the whole weighting thing with different decay curves over distance this time. And then that was multiplied by how long ago each of those houses had been sprayed. So basically, if a house that was far away was sprayed more recently, that would be a larger number. And also if a house that was close by had been sprayed like a long time ago, those might have similar numbers, but generally like closer in time and space, higher numbers, and far away in time and space, lower numbers of weighting. And so what I found from all those models is, so this was a term, this additional surrounding house sprays in surrounding houses was additional. I included it in the model in addition to the term that measured the sprays in the house itself, based on the best model that I selected in the step before. And what I found is that really including this additional term also did not improve the models at all, which is telling me that it doesn't matter what happens in the houses around you. What matters is what happens in your house and how long ago a spray happened in your house, because the models don't improve if you add this additional term. It provides no additional information. So with all that, the results from this study are that these results suggest that really, like I was saying, whatever sprays are occurring surrounding a household aren't really having an effect on the number of mosquitoes in the household. What matters is the sprays occurring in your household, in each individual household, and within those, the one that is going to actually have an effect on the number of mosquitoes in your house at any given time is the most recent one. So there's not really like an advantage of having had multiple sprays. Like you can't really be relying on a spray that happened 2 weeks ago. What matters is that you had one a week ago or as recently as possible. And so what we really, right, and this is just saying that in the same way, the reduction of Aedes aegypti in a household is determined mainly by the time since the last spray intervention in that same household. But what we don't want the message of this to be is like, oh, well then repeating sprays isn't important because this cumulative effect didn't really seem to matter. The way I interpret this is that you need to actually repeat these sprays in order to refresh the effect more often. So you can't rely on past effects that much. And these repetitions, so the frequency of sprays, should also be determined by the transmission dynamics in whatever your context is, especially thinking about extrinsic incubation period, or EIP, which is the amount of time that passes between a mosquito biting an infected person and a mosquito becoming infectious and able to transmit the virus to the next person. because say you have an infected dengue person in a house, and then you spray that house and you kill all the mosquitoes, the adult mosquitoes, but then new mosquitoes emerge from these water sites that weren't affected. And that person in that house is still infectious. So these new mosquitoes could bite this person. But it would take them 7 days or so, depending on the context, to become infectious themselves. So basically, as a vector control agency, you should be thinking about what is the EIP duration and trying to spray within that and spray for the duration of how long a person remains infectious, which can be up to 2 or 3 weeks. So basically, you would have to spray every week for 3 weeks at least to try and cut transmission. And in Iquitos, it has actually been shown that what they do there is that they like spray every week for 3 weeks, and this has been shown to have an effect on transmission reduction. But also what these results show is that really coverage is really important, because you're not getting any like spillover beneficial effect from sprays that are happening in other houses. You're really only getting an effect in the house where you're spraying. So that means that ideally you would spray all the houses, but this is very expensive and logistically complicated. So then this gets into how do we target sprays, in what areas is it more important to spray, when are we going to spray, and all those things. So, but that's not what this project gets into anymore. This is just like the discussion and contextualizing these results a little bit. So I want to thank my lab and Amy Morrison, who is also on this paper and who was so crucial in managing these projects that actually collected this data. Also, all the people in the field who performed the aspirations and the sprays and collected all the data and did all that hard work. And also my funders, which include the Pacific Southwest Veterinary Vector-Borne Disease Association and NIH, and also UC Davis School of Veterinary Medicine that's been supporting me. And I will take any questions now. Thank you.
Thank you, Ania. Very nice. Questions?
Hello, Anya. Excellent talk.
Hi.
Thank you for the wonderful
Thank
insight.
you.
I have a question. Since you mentioned coverage is sort of important based on your findings, how do you think habitat complexity or density of neighborhoods is going to influence some of the model outputs that you just talked about?
Yeah, that's a great question. Density of well, I think, okay, so the way I think about it really in this, or the way I've been thinking about it because of this study, I think when a spray occurs inside a house, it is kind of limited by the walls of that house in a way. And so I do wonder if it matters, the density, because even if your houses are in a row, if you're spraying inside a house and this is an enclosed space, there's not going to be that much exit of this spray to other houses. So even if they're very, very close by, I don't know that this spray will have an effect on other houses. And that is actually, this context is very, very close by houses. They're in rows, and that is most of urban Iquitos. So if in this context we're not seeing this effect of neighboring houses actually having sprays in neighboring houses having an effect, I very much doubt that you would see that in a context where houses are a little more spread apart. And I do think the literature supports this. I think there have been studies kind of similar that have been done in Vietnam, where I believe the houses are a little more spread apart. It's more of a like village type of context. And also they found that the effect of sprays is very limited. Yeah. Does that answer your question?
Yes. Thank you.
Other questions? I have a quick one. I was wondering if you had thoughts on whether the reduction was due to mortality or if it had to do with maybe some repellent property that was coming from the spray, given
Mm.
that it was kind of limited in the
You
breadth of the
know,
effect.
that's a good question. That's a really good question. I'm not an expert on like the chemical, you know, way insecticides work. Probably CC would be a better person for that. But two things come to mind. One of them is that there was well, two things come to mind. One of them is that in writing the discussion for this paper, I did come across some articles that suggested that cypermethrin, which is the spray that was used, does actually have a pretty long half-life inside buildings. Like after sprays, you can still find it for like pretty long periods of time, which might partially explain why we do see this like shallower decay curve than what I was actually expecting. Um, so there might be kind of some residual effect of that. But also yeah, I don't know. I like my
That's fair.
understanding is that these ULV methods, what it does is it hits the wings and then the mosquito um, dies because it's like very poisonous to it. But there might still be this residual on the walls, and I don't know that yeah, I don't know that there's a repellency effect. And yeah.
Okay. No, no problem. Just curious. Other
Yeah, I
questions?
also think
Sorry,
this
go ahead.
study wasn't, sorry, I don't think the study was like really distinguishing between indoor and outdoor mosquitoes, so it would be hard to determine that also.
Yeah.
Yeah.
Other questions for Anya? All right. Then thank you to both of our speakers. Very nice talks from both of you. And thanks to everyone who participated today. I just wanted to remind everyone that there will be another seminar next week, May 14th, also at 12 o'clock Pacific. So hope to see people there.